ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರ. ಎಜುಕೇಷನ್ ಆನ್ ಕ್ಲೌಡ್ಸ್ಗೆ ಸ್ವಾಗತ. ನನ್ನ ಹೆಸರು ನಿಮಿಲಾ. ಸೊ ಇವತ್ತಿನ ಕ್ಲಾಸ್ನಲ್ಲಿ ನಾವು ಸೆಕೆಂಡ್ PUC ಬಯಾಲಜಿನ ಚಾಪ್ಟರ್ ಆದಂಥ ಪ್ರಿನ್ಸಿಪಲ್ಸ್ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಆ್ಯಂಡ್ ವೇರಿಯೇಷನ್ನ ಕೆಲವೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಪಾಯಿಂಟ್ಸ್ ನೋಡ್ತಾ ಹೋಗೋಣ. ಸೊ ಫಸ್ಟ್ಲಿ ಇಟ್ ಇಸ್ ಜೆನೆಟಿಕ್ಸ್. ಸೊ ಜೆನೆಟಿಕ್ಸ್ ಇಸ್ ಅ ಬ್ರಾಂಚ್ ಆಫ್ ಬಯಾಲಜಿ ವಿಚ್ ವಿಲ್ ಡೀಲ್ ವಿತ್ ದ ಇನ್ಹೆರಿಟೆನ್ಸ್ ಆ್ಯಂಡ್ ವೇರಿಯೇಷನ್ ಆಫ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಫ್ರಮ್ ಪೇರೆಂಟ್ಸ್ ಟು ದ ಆಫ್ ಸ್ಪ್ರಿಂಗ್ಸ್. Now inheritance andre ಏನು? It is a process by which characters are passed on from parent to the progeny. ಅಂದರೆ ತಂದೆಯಿಂದ ತನ್ನ ಮುಂದಿನ ಪೀಳಿಗೆಗೆ ಅಥವಾ ದ ಪೇರೆಂಟ್ಸ್ ಅವರ ಒಂದು ತಂದೆ ತಾಯಿಯಂದರಿಂದ ಆ ಒಂದು ಮಕ್ಕಳಿಗೆ ಪಾಸ್ ಆಗುವಂಥ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ಸನ್ನು ನಾವು ಇನ್ಹೆರಿಟೆನ್ಸ್ ಅಂತೀವಿ. ನೆಕ್ಸ್ಟ್ ಕಮಿಂಗ್ ಟು ವೇರಿಯೇಷನ್. ಇಟ್ ಈಸ್ ಡಿಗ್ರಿ ಬೈ ವಿಚ್ ಪ್ರಾಜನಿ ವಿಲ್ ಡಿಫರ್ ಫ್ರಮ್ ಇಟ್ಸ್ ಪೇರೆಂಟ್ಸ್. ಸೊ ನಾವು ನಮ್ಮ ತಂದೆ ತಾಯಿಯನ್ನು ಸಂಪೂರ್ಣವಾಗಿ ಹೋಲೋದಿಲ್ಲ. ವಿಲ್ ಹ್ಯಾವ್ ಓನ್ಲಿ ಫ್ಯೂ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ಸ್. ಸೊ ಸಚ್ difference is called variation. So it is a degree of differences between the progeny and the parents. ಈಗ ಮೆಂಡಲ್ಸ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ಅನ್ನು ಕನ್ಸಿಡರ್ ಮಾಡಿದ್ರೆ John Mendel, the father of genetics, proposed inheritance laws, and he used garden pea as his sample, and large sampling size gave creditability to his collected data. And garden pea plant will possess certain completely opposite traits which were tall and dwarf. So the characters which he selected were one the stem height which was tall or dwarf, athwa flower color violet and white, flower position axialla terminalla anta, pod shape inflattened or constricted andre flat athwa constrict anta, pod color green athwa yellow. Next, seed shape which was round or wrinkled, seed color yellow athwa green. So true breeding pea lines were obtained by continuous self pollination over several generations, and 14 true breeding pea lines were selected as pairs and which were similar except of one character with contrasting traits. Now artificial cross pollination, which is hybridization, was performed in such varieties to obtain first hybrid generation known as the first filial progeny athwa F1 generation. Iga artificial cross pollination was performed on such varieties and this was called the F1 generation ultimately, an inheritance of one gene. So after hybridization, F1 generation so obtained resembled only one of its parents. Andre onda tall ittu illa dwarf ittu. When two plants from F1 were self-pollinated, second filial generation which was F2 generation was obtained. So illi enaitu revival of the unexpressed trait andre ಡ್ವಾರ್ಫ್ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ಸು ಈ F2 ಜನರೇಷನ್ನಲ್ಲಿ ಎಕ್ಸ್ಪ್ಲೋರ್ ಆಗ್ತಾ ಹೋಯಿತು. ಬೋತ್ ದ ಟ್ರೇಟ್ಸ್ ಟಾಲ್ ಆ್ಯಂಡ್ ಡ್ವಾರ್ಫ್ ವರ್ ಎಕ್ಸ್ಪ್ರೆಸ್ಡ್ ಇನ್ F2 ಇನ್ ದ ರೇಷಿಯೋ 3:1. ಸೊ ಇಲ್ಲಿ ಮೆಂಡೆಲ್ ಪ್ರಪೋಸ್ ದಟ್ ಸಮಥಿಂಗ್ ಇಸ್ ಪಾಸ್ಡ್ ಅನ್ಚೇಂಜ್ ಫ್ರಮ್ ಜನರೇಷನ್ ಟು ಜನರೇಷನ್ ಆ್ಯಂಡ್ ಹೀ ಕಾಲ್ಡ್ ದೀಸ್ ಥಿಂಗ್ಸ್ ಆಸ್ ಫ್ಯಾಕ್ಟರ್ಸ್. ಸೊ ಅವರಿಗೆ ಅವಾಗ ಜೀನ್ಸ್ ಅಂತ ಹೆಸರು ಕೊಡಲಿಕ್ಕೆ ಗೊತ್ತಿರಲಿಲ್ಲ. ಸೊ ಹೀ ಗೇವ್ ದೋಸ್ ಆಸ್ ಫ್ಯಾಕ್ಟರ್ಸ್. Okay, he named those as factors. So these factors contained and they carried the hereditary information and alleles. So these were slightly different form of the same factors. So factor of two alleles code for a pair of two contrasting traits. Andre yaude ಒಂದು factors ಇರಬಹುದು ಅಂದ್ರೆ genes of two alleles will code for two characteristics. ಈಗ ನಾವು ಉದ್ದ ಅಥವಾ ತೆಳ್ಳ ಇದೀವಿ ಅಂದ್ರೆ that doesn't mean we have passed on the same generations. So it is just because one of the trait is suppressed ಅಲ್ಲಿ ಅಷ್ಟೇ. ಒಂದು ಡಾಮಿನೆಂಟ್ ಇರುತ್ತೆ, ಇನ್ನೊಂದು ರೆಸೆಸಿವ್ ಆಗಿಬಿಟ್ಟಿರುತ್ತೆ. ಅದೇ ರೀತಿ ಟಾಲು ಡ್ವಾರ್ಫು ಕೂಡ. So this is how monohybrid cross, which considers only one single character which is height of the part. So ಇಲ್ಲಿ ಟಾಲ್ನ ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಎರಡು ಟಾಲ್ ಬಂತು, ಬಟ್ ಈ ಎರಡು ಟಾಲನ್ನು ಸೆಲ್ಫ್ ಮಾಡಿದಾಗ F2 ನಲ್ಲಿ ನಮಗೆ ಡ್ವಾರ್ಫ್ ಪ್ಲಾಂಟ್ ಸಿಕ್ತು. Now studying the cross breeds. So TT and TT are genotypes while the traits tall and dwarf are phenotypes. So T stands for tall trait while small t stands for dwarf trait. And even if single T is present, genotype phenotype is called tall. When T and small t are present together, then T will dominate and suppress the expression of small t. Therefore, the capital T, which is for tallness, is always a dominant trait, while small t for dwarfness is a recessive trait. Now, both capital T's and small t's are homozygous, while capital T and one small t is a heterozygous one. And from the cross, it can be found that alleles are parental pairs which will separate or segregate from each other and only one allele is transmitted to the gamete. So now gametes of the capital T will have only T alleles and gametes of small t will have only small t alleles. But gametes of capital and small t will have both the alleles. Now Punnett square. So idu graphical representation for what? To calculate the probability of all possible genotypes of offsprings in a genetic cross. So ಒಂದು genetic cross ಆದಾಗ ಅಲ್ಲಿ ಎಷ್ಟು genotypes cross ಆಗ್ಲಿಕ್ಕೆ possible ಅದಷ್ಟನ್ನ ನಾವು ಈ ಒಂದು Punnett square ನಲ್ಲಿ ನೋಡ್ತೀವಿ. So possible gametes are written on two sides, usually at top row and left columns, and combinations are represented in boxes. So idu Punnett square. Next, with the help of Punnett square, genotypic ratio in F2 generation can be found. From the above given Punnett square, it is evident that genotypic ratio of capital T, small t, and both small t is 1:2:1. So ratio is 1:2:1, or capital T, both capital T, one capital and one small t, and both small t, can be derived from the binomial expression as ax plus by whole square. Now gamete bearing genes are in equal frequency of 1. So therefore expression can be expanded as this: half t plus half small t whole square is half t plus half small t into half capital T half small t, which is 1/4 capital T, 1/2 capital T and small t, and 1/4 dwarf, which is small t's. Law of dominance, test cross, law of segregation and incomplete dominance. So Mendel's law of inheritance is idishtunu. Now based on his experiment, he proposed three laws of principles of inheritance. One, the law of dominance, law of segregation, law of independent assortment. Law of dominance and law of segregations are based on monohybrid cross, while law of independent assortment is based on dihybrid cross. Andre ಎರಡು characters anna cross ಮಾಡಿದಾಗ ಸಿಕ್ಕಿದ್ದು. Now firstly, law of dominance. According to this law, characters are controlled by discrete units called factors. As I told you, these are the genes at the present day called, which occur in pairs with one member of the pair dominating over the other in a dissimilar pair. Now this law explains expression of only one parental character in F1 generation and expression of both in F2 generation. ಈಗ ಟೆಸ್ಟ್ ಕ್ರಾಸಿಗೆ ಬಂದಾಗ cross between F2 progeny and its homozygous recessive parent. Now this cross will determine whether the dominant character is coming from homozygous dominant genotype or heterozygous genotype. Example is tallness coming from capital T or capital T and small t. Now when capital T is crossed with small t, we obtain all capital T and small t and the tall individuals in the progeny. Whereas if this heterozygous pair is crossed with the small t's, we will obtain a tall and dwarf individual in progenies. So therefore, if tallness is coming from capital T, then we obtain all tall progenies in test cross. We obtain both tall and dwarf varieties in the test cross too if tallness is coming from capital T and small t. So ee tara case 1 idu and this is case 2. So illi ಒಂದು tall dwarf cross ಆಗಿದೆ. So we are getting 2 tall and 2 dwarf trees. But ಇಲ್ಲಿ ಒಂದು tall ಮತ್ತೊಂದು dwarf ಪೇರ್ ಆದಾಗ ಇದು ಡಾಮಿನೆಂಟ್ ಆಗ್ಬಿಟ್ಟು ಎಲ್ಲವೂ ಟಾಲೇ ಸಿಗುತ್ತೆ ನಮಗೆ. Okay, so now coming to law of segregation. So e law ಏನು ಸ್ಟೇಟ್ ಮಾಡುತ್ತೆ? It states that two alleles of pair segregate or separate during gamete formation such that a gamete will receive only one of the two factors. So ಇದು ಹೇಗೆ segregate ಆಗುತ್ತೆ ಅಂದರೆ ಎರಡು traits ನಲ್ಲಿ ಯಾವ್ದಾದ್ರು ಒಂದೇ ಒಂದು factor ಬರೋ ತರ it is going to segregate or get separated. And in homozygous parents, all gametes produced are similar, while in heterozygous parents, two kinds of gametes are similar and one are dissimilar. Now incomplete dominance. So in incomplete dominance, F1 generation has phenotype which will not resemble either of the parents, but it is a mixture of the two. Example, flower of the color in dog flower. So it is snapdragon agata where RR is the red flower. So now coming to law of segregation. So e law in state ಮಾಡುತ್ತೆ the two alleles of a pair segregate or separate during gamete formation in such a way that only one of the factors between the both are expressed. So ಇಲ್ಲಿ homozygous ಬಂದಾಗ homozygous parents ನಲ್ಲಿ all of the gametes will be similar, whereas in heterozygous parents two kinds of gametes are produced in equal proportions. So next coming to incomplete dominance. So incomplete dominance, so F1 generation has a phenotype which will not resemble either of the two parents, but it will have the characteristics of both the parents. Example, flower color in dog flower, which is snapdragon. So ಇಲ್ಲಿ red flowers ಮತ್ತೆ white flowers ಸೇರಿ pink flowers ಅನ್ನ produce ಮಾಡತ್ವೆ. Now here genotypic ratio remains the same as in Mendelian's cross, but phenotypic ratio will change because there is no complete dominance shown by R. So it is incomplete dominance ಅಥವಾ codominance, as I told you. Now phenotypic ratio na ನೋಡಿದಾಗ it is 1:2:1, wherein red, pink, white ಮೂರು denote ಆಗಿದೆ. Genotypic ratio ಬಂದರೂ ಕೂಡ it is the same, wherein two reds, one red, one white and two pinks are distributed here. Next, what is dominance? So a diploid organism produces two copies of genes which will not be identical and they may have minor alternates. Ega suppose a normal gene produces a product P, then alternated version of it must produce a non-functional product P, athava there should be no product formation only. Okay. Now original gene is said to be dominant while the modified gene is recessive one. ಈಗ law of segregation and codominance. So codominance andre ಏನು? In codominance, F1 progeny will resemble both the parents. Example, ABO blood group in human beings. So ಇಲ್ಲಿ ABO blood group ನಲ್ಲಿ ಏನಾಗುತ್ತೆ? They are controlled by gene I, wherein gene I has 3 alleles, IA, IB and only I. So a person possesses any of the two of these 3 alleles. So ಇಲ್ಲಿ IA and IB dominate over I. But with each other IA and IB are codominant. Okay. So ಇಲ್ಲಿ IA and IB contain both A and B type of sugars. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ? I ಅಲ್ಲಿ ಯಾವುದೇ ರೀತಿಯ sugars ಇರೋದಿಲ್ಲ. So this is how allele from parent 1 and parent 2 and it is going to get transferred to the offspring. So allele 1 from parent 1 will be IA and IA, IA ಅಥವಾ IB, IA and IB IA, IB IB and IBI and both I's. So ಈ ರೀತಿ ಆದಾಗ genotype ನೋಡೋಣ. So genotype ಎರಡು IA ಆಗುತ್ತೆ ಅಥವಾ AB ಆಗುತ್ತೆ, A and only I ಆಗುತ್ತೆ, again AB, both B's, ಒಂದೇ ಒಂದು B and ಯಾವುದೇ ಇಲ್ಲದ ರೀತಿ ಆಗುತ್ತೆ. So this will become a blood type of A positive, A, AB, A again, AB, B, B and O. So ಇಲ್ಲಿ positive or negative will depend on the RH type of the offsprings. Now coming to multiple alleles. So ಇಲ್ಲಿ when more than 2 alleles control a character as in human blood groups O, multiple alleles are used in the populational studies. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ? Multiple alleles ಒಂದು blood group ಅನ್ನ control ಮಾಡ್ತಾ ಹೋಗುತ್ತೆ, ಒಂದು character ಅನ್ನ. Now inheritance of two genes, that is dihybrid cross and law of independent assortment. So ಇಲ್ಲಿ inheritance of two genes and it is dihybrid cross. So in dihybrid cross we consider two characters like seed color and seed shape. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ? Yellow color and round shape is dominant over green color and wrinkled shape. So ಈ ರೀತಿ ಇದು Punnett square. Okay. ನಾವು ಫಿನೋಟಿಪಿಕ್ ರೇಷಿಯೋ ಅಂತ ನೋಡಿದಾಗ 9:3:1 ಬರುತ್ತೆ. ಇಲ್ಲಿ ರೌಂಡ್ ಯೆಲ್ಲೋ 9 ಇರುತ್ತೆ, ರೌಂಡ್ ಗ್ರೀನ್ 3 ಇರುತ್ತೆ, ವ್ರಿಂಕಲ್ಡ್ ಗ್ರೀನ್ 3 ವ್ರಿಂಕಲ್ಡ್ ಯೆಲ್ಲೋ 3 ಇರುತ್ತೆ, ವ್ರಿಂಕಲ್ಡ್ ಗ್ರೀನ್ 1 ಇರುತ್ತೆ. So law of independent assortment is what it states. So when two pairs of traits are combined in a hybrid, one pair of character is going to segregate independent of the pair of other character. Now in dihybrid cross between two plants having round yellow and wrinkled green seeds, 4 types of gametes, that is capital R, capital Y, capital R, small r, capital Y and both small r, small y are produced. So each of these will segregate independent of each other and each having a frequency of 25% of total gametes produced. Now coming to chromosomal theory of inheritance. So this is just a rediscovery of Mendel's work. So it remained unrecognized for several years because of the following reasons. Why? One, the lack of communication and publicity in those days, and his concept of factors, genes are discrete units which did not blend with any of the accepted of light of variations occurring continuously in nature. ಈಗ Mendel's approach to explanation of biological phenomenon with the help of mathematics was also not accepted. ಸೊ ಒಂದು ಬಯೋಲಾಜಿಕಲ್ ಕಾನ್ಸೆಪ್ಟನ್ನು ಮ್ಯಾಥಮೆಟಿಕಲ್ ಆಗಿ ಎಕ್ಸ್ಪ್ರೆಸ್ ಮಾಡಿದಾಗ ಅದನ್ನು ಎಕ್ಸೆಪ್ಟ್ ಮಾಡ್ಕೊಳ್ಳೋಕೆ ರೆಡಿ ಇರಲಿಲ್ಲ. ನಾವು ಇನ್ 1990 3 scientists Hugo de Veres, Correns and von Tesmack independently rediscovered this Mendelian's work. ಸೊ ಇವ್ರು 3 ಜನ ಒಟ್ಟಿಗೆ ಕೆಲಸ ಮಾಡಿಲ್ಲ. ದೇ 3 ಸೆಪರೇಟ್ಲಿ ಡಿಸ್ಕವರ್ಡ್. ನಾವು ಕ್ರೋಮೋಸೋಮಲ್ ಥಿಯರಿ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್. ಸೊ ಇನ್ 1900 ಡ್ಯೂ ಟು ಅಡ್ವಾನ್ಸ್ಮೆಂಟ್ ಇನ್ ಮೈಕ್ರೋಸ್ಕೋಪಿ ಕ್ರೋಮೋಸೋಮ್ಸ್ ವರ್ ಆಲ್ಸೋ ಡಿಸ್ಕವರ್ಡ್. So ಇಲ್ಲಿ Sutin and Baveri discovered the behavior of chromosomes which was parallel to the behavior of genes. So chromosomes and genes both occur in pairs, which is 2 alleles of a gene paired and located on the homologous sites of homologous chromosomes. ಈಗ Sutin Baveri further proposed that it was the pairing and separation of pair of chromosomes which ultimately led to the segregation of a pair of characters which they are going to carry. ಇಲ್ಲಿ union of knowledge of chromosomal segregation with the Mendelian's principle constitutes chromosomal theory of inheritance. Now dihybrid crosses in Drosophila to study linkage of recombination. So linkage and recombination andre ಏನು ಅಂತ ನೋಡೋಣ first. So Thomas Hunt Morgan discovered the basis of variations that sexually reproduction was produced. So he worked on fruit flies, which is Drosophila melanogaster. Now, they were suitable to grow on a synthetic medium in laboratory. Why was he choosing these antandre? Now, these life cycles would have got completed in 2 weeks and single mating will produce many progeny flies and clear differentiation of sexes which is easily distinguishable between male and female, and heredity variations were clearly visible with lower microscopes. Now, Morgan's experiment. So, dihybrid class was carried out on fruit flies. So yellow-bodied white-eyed females were crossed with brown-bodied red-eyed males. Now F1 progeny was obtained which was intercrossed. Now F2 progeny was obtained and F2 ratio was observed. So F2 ratio was observed to a significantly different form which is 9:3:3:1. So ಇಲ್ಲಿ explanation of Mendelian ratio ಆಗ್ತಾ ಹೋಯ್ತು. So now genes involved are located on X chromosomes. Okay, this is what it states first. Now when 2 genes are located on same chromosomes, the proportion of parental gene combinations was higher than those of non-parental genes. ಈಗ linkage. ಈ linkage, it is a physical association of genes on the chromosomes and recombinations. So this is how it is going to take place. Now Alfred Struveran utilized the knowledge of frequency of gene recombination as a measure of physical distance between 2 genes to map their positions on chromosomes. So idana gene mapping antivi. Now in this way genetic maps were prepared which were extensively used today for sequencing projects as in HGP, Human Genome Project. Now sex determination in various animals which include human, males and females heterogamety, that is sex determination. So ಇಲ್ಲಿ Henking discovered the genetic chromosomal basis of sex determination by working on insects. So he observed specific nuclear structures during spermatogenesis in insects and he named these structures as X bodies. So ಇಲ್ಲಿ chromosomes involved in sex determination are called the sexual chromosomes, which is the 23rd pair chromosome. And while the others are called the autosomes, the remaining 22. So ಇಲ್ಲಿ XO type of sex determination ಏನು? Other than autosomes, one X chromosome is present in all the insects. So some sperms contain X chromosomes while the other do not. Now eggs fertilized by sperms have X chromosomes and they become females. So females will have 2 X chromosomes and eggs fertilized by sperms not having X chromosomes will become males. So males will have only 1 X chromosome. Example of the organism with XO type of sex determination is insects. ಈಗ XY type. So ಈ XY type humans ಅಲ್ಲಿ ನಾವು ನೋಡ್ತೀವಿ. Males have X chromosomes and counterpart of Y chromosomes which is distinctly smaller and hence males are XY. Females have pair of X chromosomes and hence females are XX. Example of organisms with XY type of sex determination are humans and Drosophila. Now coming to male heterogamity, XO and XY types of sex determination are the best examples of male heterogamity. So illi XO type, some gametes have X chromosomes while some are without X chromosomes, and in XY type some have X chromosomes while some gametes have Y chromosomes. And female heterogamity, so illi ZW type of sex determination is an example of female heterogamity. So illi ZW, female has Z and one W chromosome while male has a pair of Z chromosomes. Now coming to mutation, pedigree analysis and genetic disorders. So mutation andre eno, it is a sudden change or alternation of the DNA sequence which results in change in genotype and phenotype of the organisms. So illi enagatte, DNA helix is going to run in a chromatid, and hence any change which is insertion or deletion in the DNA sequence will affect the chromosomes. Illi point mutation antagatte. So mutation arising due to change in single base pair of a DNA is called sickle cell anemia. So now coming to frame shift mutation. So this is a type of mutation which is occurring due to addition or deletion in the DNA sequence. Now next coming to mutagens. So these are chemical or physical agents which will lead to mutations. Example, UV radiations. Next coming to pedigree analysis. So pedigree analysis, it is an analysis of inheritance of traits in several generations of a family. Now a particular trait under study is represented in a family tree. Now by using pedigree analysis, inheritance of specific trait, abnormality or disease can be traced easily. Now DNA, it is believed to be the carrier of genetic information which will pass unaltered from one generation to the other. Now mutations occasionally will alter this genetic material, and genetic diseases are believed to be associated with these alternations. Now standard symbols in pedigree analysis are as follows. So illi these are the symbols which are used, and this is a pedigree chart which is represented. Okay, male lige, female lige, and sex unspecified, affected individuals, mating between relatives, mating above and children below, and parents with male child affected with disease, and 5 unaffected offsprings. Now chart A will represent inheritance of autosomal dominant trait as in muscular dystrophy. So now chart B will represent inheritance of an autosomal recessive trait as in sickle cell anemia. Now genetic disorders. So include Mendelian disorders and chromosomal disorders. So Mendelian disorders yavdu? They are characterized by mutation in a single gene, and their mode of inheritance will follow the principles of Mendelian genetics. And Mendelian disorders can be either autosomally dominant, which is muscular dystrophy, and autosomal recessive, which is sickle cell anemia, and sex linked, which is hemophilia. Now sex linked recessive disease is hemophilia. Illi transmission from unaffected female to male progeny is happening. Now females will act as carrier of the disease but rarely suffer from hemophilia, because for a female to become hemophilic, the mother should be carrier and father should be hemophilic. Now in the disease, protein involved in blood clotting is affected. Now sickle cell anemia. So it is an autosomal recessive disease wherein transmission from parent to offsprings when both are carriers of the disease is going to happen. Now pairs of alleles HBA and HBS will control the expression of this disease. So HBA and both HBA and HBA should be normal here. Athwa HBA and HBS should be carrier of the disease, and HBS and HBS should be diseased. Now cause of this disease is change in gene causing the replacement of GAG and GUG, which will lead to the substitution of the glutamine molecule by VAL at 6th position of the beta globulin chain present in the hemoglobin. So this mutant hemoglobin so formed polymerizes at lower oxygen tension. This is ultimately going to result in the change of shape of the RBC to sickle like. Okay. Now coming to phenylketonuria. So it is an autosomal recessive disease, and phenylalanine tyrosine is a main causative enzyme. So the enzyme responsible for this conversion is going to get mutated, and now phenylalanine will accumulate, and then this phenylalanine will become phenylpyruvic acid, and it is going to accumulate in the brain causing mental retardation. And next, coming to phenylpyruvic acid, which will also get excreted through urine because kidneys will be very poorly able to reabsorb those. Now, chromosomal disorders. So, total number of chromosomes in humans is 46, which is 23 pairs. So, total 23 pairs, it is 22 pairs of autosomes and 1 pair of sex chromosomes. Now, monosomy is the lack of any one of the pair of chromosomes, and trisomy is inclusion of additional copy of chromosomes, and aneuploidy, which is loss or gain of chromosomes because of the failure of secretion of the chromatids. Now coming to Down syndrome. So cause is the presence of additional copy of chromosomes in 21. So trisomy of 21 will happen here. Affected individual will have short stature, small round head and furrowed tongue and partially opened mouth, and palm will crease here, and congenital heart disease and mental retardation is seen. Now coming to Klinefelter syndrome. So cause of this is additional copy of X chromosome, which is 47th chromosome, which will be of XXY type, and affected individual will have overall masculine in development with gynecomastia, and the individuals will become sterile. Now Turner syndrome. So absence of X chromosome, which is 45th chromosome XO, and affected females will be sterile in this, and rudimentary ovaries can be seen, and then secondary characters will be absent. Thank you.